Hello dear kiddos, hope you all are doing good. So today we are going to continue with part 6 mock question paper 2025. Let us begin without any further delay. Here goes the very first question. In a monomeric antibody molecule, how many intra-chain disulfide bonds are present in light chains and heavy chains respectively? 10 to 8, 4, 4, 8, 12, 4 respectively. Come on, think and answer the question. Time starts 3, 2, 1 right now. Alright, so the answer is 4 and 8. How? So if you see, there are 2 light chains and 2 heavy chains, right? In a monomeric antibiotic molecule, if you see, there are 2 light and 2 heavy chains. And if you look at light chains, how many intra-chain disulfide bonds are going to be there in each chain? 2, right? Yes, so 2 into 2, it becomes 4. So there are 2 heavy chains and each chain is going to have 4 intra-chain disulfide bonds. So 2 4s are 8. So it's going to be 4, 8. I have given you the explanation here. So 2 light chain, 2 heavy chain. Each light chain will have 2 intra-chain disulfide bonds. So it's 4. And heavy chains, each chain will have 4 intra-chain disulfide bonds. So the total number of heavy chains will be 8. So 4, 8 is the answer. In rDNA technology, rDNA means DNA with a piece of RNA, DNA with a piece of foreign DNA, DNA which takes part in recombination. DNA does not associate with any foreign gene. Your time starts 3, 2, 1 right now. This is a very simple topic, right? You're learning what is this right from your grade 9, grade 10. Yes. So first of all, rDNA means recombinant DNA, the DNA with the piece of foreign DNA. Yes, it stands for recombinant DNA. It is made by joining DNA from 2 different sources. Usually a foreign gene is inserted into a host gene, which is like a plasmid, right? Next one. Adenosine deaminase deficiency may be cured permanently by administering adenosine deaminase promoters in the blood of the patient, introducing bone marrow cells in the patient, isolating gene from marrow cells producing ADA and introducing these genes into cells at early embryonic stage, enzyme replacement therapy. What do you think? How can adenosine ADA, we say, deficiency can be cured permanently? Your time starts 3, 2, 1 right now. Come on, my dear kiddos, I want everybody to answer it. Yes. So the answer is nothing but isolating gene from the marrow cells and producing ADA and introducing those genes early at the embryonic stage. That is how we can prevent ADA deficiency. Right? The gene has to be introduced at embryonic stage. Therefore, it becomes part of the body cells and it will eliminate the deficiency. Next one. Choose the odd one with respect to nucleosides. Adenosine, cytosine, guanine and thymine. Your time starts 3, 2, 1 right now. Yes, the odd one out they are asking you. So children, basically the answer is cytosine because if you see adenosine, AGT, guanine and thymine, these are nucleosides because they have a nitrogenous base along with the sugar. Whereas nitrogenous cytosine, if you see, it's just the nitrogenous base, so it's not a nucleoside. Select the incorrectly matched pair from the options given below. Genetically engineered insulin prepared by Eli Lilly. RNAi, silencing of mRNA with the help of dsRNA, double-stranded RNA. Rossi, transgenic sheep introduced alpha-1 antitrypsin. Golden rice, source of vitamin A. Your time starts 3, 2, 1 and you have to pick the wrong one, incorrectly matched one. Your time starts now. Come on kiddos, you can do it. Okay. Yes, so the answer is Rossi. So do you think, what do you think is the error here? Rossi is a transgenic cow, not a sheep, right? Yes, and it was developed to produce human protein enriched milk, especially alpha lactalbumin, which is good for babies. And Eli Lilly made genetically engineered insulin, and RNAi is silencing of mRNA using dsRNA. Golden rice is rich in vitamin A, which is beta carotene. Yes. In the experiment performed by S.L. Miller in 1953, he observed that the formation of P can form a polymeric compound by formation of blank with each other. Choose the option. Glycosidic bond, peptide bond, phosphodiester bonds, phosphoester bonds. Your time starts 3, 2, 1 right now. Yes. So the answer is peptide bonds, right? Yes. S.L. Miller did an experiment to show how life's building blocks could form at an early Earth. He observed the formation of amino acid. The amino acid can join together to form proteins. They do this by forming the peptide bonds. What is the bond between the amino acid which forms protein? Peptide bond, right? So the answer is option B. The parts of human female reproductive system along with a pair of mammary glands are integrated structurally and functionally to support the process of pregnancy, parturition and childcare, fusion of spermatozoa and ovum, release of secondary oocyte from primary sex organ. Select the correct option. A only, B and C only, A, B, C and A, B and C, C only. What do you think? Your time starts 3, 2, 1 right now. The parts of human female reproductive system along with pair of mammary, they are integrated structurally and functionally to support all of these processes, right? First is all about pregnancy, parturition and childcare, right? And then talking about fertilization, right? And then release of secondary oocyte is ovulation. So all of these are structurally and functionally supported by the human female reproductive system and also the pair of mammary glands. True. Consider the following flowchart and select the correct statement with respect to hormonal regulation of spermatogenesis in human. In 2024 paper, previous year paper, you can see this question. X is a steroid hormone that stimulates the release of two gonadotropins. Z is the amino acid derivative that stimulates the secretion of androgen. Y is a steroidal hormone that stimulates the secretion of some factors which help in process of reduction division. Y and Z are trophic hormones. So what do you think is very true regarding X, Y and Z? Your time starts. 3, 2, 1, right? Now, you have to just select the correct statement with respect to spermatogenesis, right? So children, Y and Z are trophic hormone. That is the true statement. Why others are wrong? Look at this now. X is nothing but gonadotropin releasing hormone released by the hypothalamus. That's it. It will go and stimulate the adenohypophysis is nothing but the anterior pituitary. Correct, because anterior pituitary has so many hormones being stimulated, whereas the posterior just two. What are the two? What are the two? Oxytocin and vasopressin, right? That's it. Whereas anterior pituitary has so many hormones being released, but here in case of Y and Z, Y is stimulating Leydig cells, means Y is luteinizing hormone. Z acts on the Sertoli cells, means it's nothing but the follicle stimulating hormone. These two hormones are trophic hormones. They are not steroid. What is meant by trophic hormones? Because they stimulate other glands or cells to perform the function, where luteinizing hormone is going in, stimulating Leydig cells to release testosterone. Right? Yes. So likewise, FSH is going and stimulating the Sertoli cells. So keep that in mind. How many of the methods given in the box below generate generally do interfere with the sexual drive or desire or the sexual act of the user? LNG20, Walls, Haley condoms, Progestasert, Multilode 375, select the correct option. 5043 is how many of the given do you think interfere with the sexual drive, desire or the sexual act? Your time starts. 3, 2, 1 right now. Alright. Student, the answer is 0 because none of them will interfere with the sexual drive, desire or sexual act because all of these are contraceptive devices which will prevent the pregnancy, which will prevent fertilization from happening. Right. They have nothing to do with the sexual drive, desire or the sexual act. Correct? Yes. Choose the incorrect statement. Myosin head contains ATPase and has ATP binding sites. Repeated contraction and relaxation of the skeletal muscles may lead to fatigue. The cells of human body exhibit amoeboid, ciliary and muscular movements. Sarcomere is the anatomical unit of muscles. Which do you think is the wrong statement here? Your time starts. 3, 2, 1 right now. Come on, kiddos. You can do it. Yes. Do you think sarcomere is an anatomical unit? It's the structural and functional unit of the muscles. Right. That's why it's wrong. All other statements are very true. Yeah. Yeah, see, the structural and functional unit of muscle, sarcomere, right? Whereas myosin head has ATPase that binds to ATP, muscle fatigue happens with repeated use, and the body shows amoeboid, ciliary, and muscular movements, right? I think that was the last question. I think we completed quickly. That's great. Fine. Children, I hope these questions are useful for you. For more such videos, please do like, share, and subscribe. And all the best for your exams, right? Yes, yeah, you have to shine better and keep following more channels there. And one more thing, children, there are PYQ videos also. Go through that. 2023 complete paper is done. 2024 complete paper is done. What we are doing right now is mock series, right? So please go and watch those videos as well, which will be very helpful for you. Okay. Yes, all the best. Tata. Take care.